thank you also for the organizing committee to give me the opportunity to present today. So I do have disclosures associated to this talk. I have decided, and I was asked to by the organizing committee, to focus a bit on the approval-relevant news in Switzerland. So we do have a new anti-CD20 antibody, which was approved in February. We do have approved newly the biosimilar natalizumab. But I was especially also asked by Lara Diem, as the president of the committee, to focus on the situation of BTK inhibitors. A little bit the elephant in the room after the talks we heard before. What can we do against progression? And this is really what we all hoped, that I have the solution today. However, during Christmas, around Christmas time, there were news that at least raised some question marks, and I will allude to that a bit later. So, coming to the new anti-CD20 antibody, ublituximab. So what is the rationale to develop a novel anti-CD20? It's very clear that these antibodies would differ in terms of sequence, being human or being more murine, different epitopes, but most importantly, also especially in functionality. There are different ways of how you can deplete a cell, a B cell specifically, and all the antibodies differ a bit. What we can say is that ublituximab has a higher potential to go for antibody-dependent cellular cytotoxicity, here shown on the right-hand side. This is not unique, but there's a specific sort of preponderance of this antibody. And this kind of cytotoxicity will be exerted via specific receptors, the Fc gamma receptors. Why is this important? Because ublituximab is glycoengineered, meaning carbohydrate residues on the antibodies have been modified to enhance this specific binding to the Fc gamma receptors. And that you can see on the left-hand side, where it's quite clear ublituximab really has an increased binding affinity. And that holds true for the blue and the orange bars on the left-hand side. What is this? The blue ones are the high affinity receptors, but there are also SNPs which code for a variant which then would encode for a lower affinity Fc gamma receptor, meaning that both sort of variants of the receptor would be addressed by this glycoengineering. Is that functionally relevant? We don't know in MS. However, and at least in NMOSD, there's some indication. So when you compare NMOSD treated with rituximab, this is around 100 patients or so, it becomes quite clear if you have a variant which encodes for this lower affinity Fc gamma receptor, then the likelihood to develop one or more relapses under rituximab is much higher. So yes, it may also well be translate into MS. Now here's the data, the 5-year data now from ublituximab. It's a typical design, 2-year course study against an active comparator, teriflunomide, and then the patients were switched from teriflunomide to ublituximab for the remaining time. On the left-hand side, what you can observe is the relapse rate, a clear reduction in the relapse rate, not surprisingly, which is carried over also in the long-term follow-up. And on the right-hand side, the 24-week confirmed disability progression. And also here you see a divergence of the curves. However, this is not true during the first 2 years. But only if you look at the whole 5 years, there's an indication that if you have been treated with that substance earlier on, that there may be a long-term benefit, however, with all the caveats of a open-label design. There are other pragmatic sort of differences which I have listed down here. All over, this led in February this year to the approval of that substance for active MS, as we know, with either clinical or paraclinical MRI parameters. Switching the gears, natalizumab biosimilars. You know that generics and also biosimilars have a large potential. So we are thinking pharmacoeconomically that especially biosimilars could save the system around 30% of the costs. So we do have a newly approved natalizumab biosimilar. What does the approval process for a biosimilar look like? On the left-hand side, you see the normal approval process for the reference medicine. On the right-hand side, the pyramid, the flipped pyramid, the biosimilar. It's quite clear for the reference, you need a clinical study. You need clinical phase 3 trials. That's really the top of the pyramid there. This is really different with the biosimilar. There, you have an extensive physical, chemical, structural, and functional characterization as the basis of the approval process to really show that the biosimilar matches in all these aspects the reference originator substance. And then the clinical part is much lower and much smaller in that you are supposed to choose endpoints which demonstrate no clinically meaningful differences. And that was indeed shown in this phase 3 trial, which is different, as you can see, because it is relatively small, 260 patients, and did have an MRI endpoint of new active MRI lesions, which are the same between the biosimilar and the originator substance. Now, we do have similarities, but also we do have uncertainties when it comes to practical use, and that's shown in here. The uncertainties refer to the risk stratification for PML when using the typical anti-JCV ELISAs. So this is a study from England, the UK, about 500 patients. On the left-hand side, you see patients which were treated with Tysabri and tested with the corresponding stratified test. These patients were then switched to Tyruko, which is the biosimilar, and they were tested with a different ELISA. This is logistically, because of payment, one has to do it. And this assay is then called ImmunoWell. So what did you see? It's quite clear if you have two different assays, you will have divergent results. However, in clinical practice, it looks like pretty substantial in that about 50% from the stratified negative patients were reported as being positive with the ImmunoWell assay. Or in other words, also those with low positive antibody index values with stratify were reported high antibody positive in the ImmunoWell. And you can easily anticipate these are really the thresholds where it comes to action for us clinicians, right? Where we would change our monitoring, where we would even consider switching the agents. Now, this is really unclear what to do. It's not a better or worse assay, it's just a different assay. It's quite clear if you do something like that and do not have a uniform testing platform. Whenever it comes to these uncertainties, and especially in the UK, many patients especially were really unsure how to move forward, and therefore it's very important to have a clear consensus. And under the guidance of Ilyas Jelcic, who is also here in the room, also the Swiss MS Society developed in analogy to the UK sort of a consensus. So this situation, if you are Tysabri pre-treated, if you switch to Tyruko, if you have been negative and if you remain negative, is easy. You continue with the normal procedure. The other extreme, so you are known positive, but you have a strong increase of the antibody index. This is also somewhat easier in that you really would increase the monitoring and consider switching. However, when you come from negative and are here somewhat low positive, that's really difficult because you really don't know, is this an issue of the assay or is this true biology? And here, like the UK physicians, we consider different strategies. You can all read them up in the consensus statement that we're having. Now, coming to the elephant in the room, this photograph is probably the most often shown photograph during neuroimmunological congresses over the past 2 years. This is Ogden Bruton, a pediatrician who identified clinical cases of A gamma globulinemia with a severe block in B-cell maturation. And it's quite clear that one could pharmacologically exploit that in inhibiting the cytoplasmic enzyme, which is relevant for B-cell maturation, also in autoimmune disease. Is that relevant also in MS? So the idea goes like that. We believe that we do have peripheral B cells, which would be attacked by the BTK inhibitors, and also myeloid cells, which would be addressed by the BTK inhibitors. And the peripheral ones, would somehow be associated with relapse biology. But however, as we have heard very elegantly this morning by the 2 talks, we also do have CNS compartmentalized inflammation, which is proposed to be associated with progressive biology. And this is shown in here. So this is the lesion rim that we heard of earlier this morning, which clearly stains for BTK. This is increasing the stain. This is Western blot then, especially in chronic disease. And yes, it's also co-localized the staining with microglial markers. So really the hypothesis is we will want to have a CNS penetrant, ideally non-depleting small molecule, which will both impact on the relapsing and the progressive biology while, because not depleting, being safe. So this is the whole rationale beyond the development programs of the different companies. Do we have some indication, do we have some support that there's really an effect on the CNS compartmentalized inflammation? Yes and no. So on the left-hand side, you have the phase 2b long-term follow-up. Small cohort, quite short so far. Yes, there may be an indication that there's a decrease over the long-term follow-up with tolebrutinib in the decrease of slowly evolving lesions. When you look at the paramagnetic rim lesions, you see a mixture of everything. Mostly the patients remained stable with their paramagnetic rim lesions. However, there were occasional patients who had an increase, but also patients who had a decrease of paramagnetic rim lesions. So I would argue here the story is not really entirely clear. However, there may be an indirect something. Christina introduced that, the threshold of more than 4 paramagnetic rim lesions. That may really be a factor which is associated with a stronger efficacy on disability outcomes with tolebrutinib. So those patients who had 4 or more paramagnetic rim lesions at baseline were really the ones who benefited best in the phase 3 trials in terms of progression outcome parameters. Now, how different are the BTK inhibitors? Is it really worthwhile to go for different substances, 5 different substances? Potentially, yes. Because the mechanism of action is different. So we have like a reversible versus a irreversible covalent binding. The selectivity for binding to BTK and potentially via off-target effects to cause side effects may be different, but also the CSF exposure. Now, CSF exposure, which is very often used as the ideal something, is it really the ideal something? So in our group, we're looking at ways to enhance penetration of large molecules, namely antibodies, into the CNS. For example, here on the left-hand side, by generating bispecific antibodies, which dock to a receptor which facilitates the crossing over the barrier. And what you can easily observe in here, yes, that works when you look at the concentration of the antibody in the parenchyma in comparison to the non-bispecific antibody. However, when you look at the CSF, there's absolutely no difference. So really, CSF is not ideal. And the same we see when we, for example, this is another approach that we're using, do a targeted, region-targeted application intranasal antibodies. And again, here we do see clear concentration peaks in the tissue. However, regardless of which time point, we just don't see anything in the CSF. So be aware of CSF, which is clear. So where are we standing? Enthusiasm was quite big because around here, ACTRIMS 2024, there was clear efficacy of tolebrutinib in non-relapsing secondary progressive MS. So secondary progressive MS patients with no relapses over the past 2 years. However, as I mentioned, in early December, there was the press release that a parallel study on PPMS is negative. And then exactly on 24th of December, FDA released a letter where the requested indication, non-relapsing SPMS, was at that time neglected. Why is that? So there is a clear response letter by the FDA. So they argue the benefit risk is not ideal in that there's a high risk of severe drug-induced liver injury. And that if you see it preclinically, then post-clinically, post-marketing, this may even increase. There are uncertainties about really the optimal benefit in which group because we still had focally active patients in that study that at baseline did have 13% of MRI activity. And what I personally find quite interesting, whereas PERA for us is a given, is like a natural constant, FDA, and not talking about like the wisdom of US agencies currently, but here I find this interesting in that they really question and caution the concept of PERA. So what is the deal right now? Currently, this is the ultimate decision is postponed to the end of Q1 this year. FDA requests additional data really to characterize the current safety programs running for safety monitoring on the incidence of drug-induced liver injury. So what are we talking about? What kind of liver injury? If you look at that, it doesn't look too amazing. However, the devil is in the detail. This is really what we're looking at. This constellation is biochemically the High's law. This is not coined by himself, but by an FDA person long time ago. So if you have a combination of transaminases, upper 3 times upper limit of normal, and the total bilirubin above 2 times upper limit of normal, and no other causes, then this signal is believed to be at least associated with a 10 times higher risk of liver failure or mortality even, leading to transplantation. So that's really a strong indicator for the FDA and also the EMA. So where are we with the other substances? Shortly before the turn of the year, we did have a press release of fenebrutinib with one relapsing trial. We did see a efficacy against teriflunomide in relapse outcomes. And in the PPMS trial, there appears to be also efficacy when compared to Ocrevus. However, in a non-inferiority design. I will come back to that later. No more details are clear in here. So this is my personal where we are right now. And this is not entirely correct. Because what I'm doing in here is I'm lumping clear data, published data with press release. I'm sort of lumping different controls. I'm sort of bit of lumping different outcome parameters. However, to make things easy. So ibrutinib is negative in relapse outcomes, in MRI outcomes, referring to focal activity and brain volume. When it comes to tolebrutinib, then okay, it's quite clear. It may make sense when it comes to relapses. Everything is negative. It looks like one substance, the fenebrutinib, at least in one trial, may be positive. We will see. Here it becomes much more difficult. So when we look at progression parameters, then yes, 2024, we did see in relapsing trials and in non-relapsing SPMS efficacy. However, with the same substance, with potentially the same kind of pathomechanism of progression, this was negative. Here may be new hope. However, you have to be aware the trial, the PPMS trial, FENtrepid, was designed as a superiority trial. Now we're reading about non-inferiority, so we really would have to see the final data. And then it comes to imaging. Imaging is always a conundrum for me, so I'm glad that Christina is there to explain it to me. However, this I really don't understand. So we do have a dissociation between T1 and T2 with the BTK inhibitors, in that, yes, both are mostly negative, but we really even see a divergence, a numerical increase with the BTK inhibitors of T1 lesions and a decrease of T2 lesions. I don't understand that. And what I also do not understand is a clinical efficacy on progression parameters despite the lack of efficacy on brain volume. I still have 9 minutes, Pasquale. It's shown in here. But I will hurry up. I know that. So what I'm trying to say, rather than giving you clear answers, I'm afraid I have at least smaller to intermediate question marks in my brain. I hope next time we will be able to answer that. Coming slowly to an end, where are we right now? We don't know with the BTK inhibitors. But nevertheless, I mean, we also deal with a subset of patients where we just are weaponless. And Menno alluded to that. And I'm very grateful that the second part really was also about cognition and fatigue, because this is a typical patient that was referred to us as a potential candidate for CAR T-cell trial. Robert, who's also in the room, is very successful in setting that study program up in our side. And that young female was stable neurologically, but neuropsychologically, she had increasing fatigue and cognitive decline, despite a combination of anti-CD20 and anti-IL-17. So where are we? Why are we so bad in tackling this? Because maybe our concepts are not proper, and maybe we should have to switch our concepts. Quite likely, fatigue is multifactorial. There may be genetic, immunological, functional and structural abnormalities, and of course, behavioral abnormalities. And over time, they may not behave similarly and not in parallel. So there may be different windows of opportunity to treat in different ways. And it's always good to show a picture of the chairwoman when one talks. So I brought along this. Iris at our side, together with Naomi, in an ongoing clinical study, they were successful in trying a different approach, where they take neuroeducation modules quite extensively with a specific rationale, combine it with mindful-based techniques, and look at here cognitive outcome parameters and subjective and objective outcome parameters. Here, really, there is an efficacy, so we're really looking forward to see more of that. Coming to an end now, Pasquale, still more than 6 minutes. Yeah, I think we do have a novel kit on the block, the novel CD20, which is approved for relapsing. So the obvious question really is it like, for example, like what we thought about the direct anticoagulants, right? Because of a different mode of action, shall we switch laterally? We just have no data, so we really have to understand what the true potential of this antibody is. When we talk about switching to biosimilar natalizumab, I think this is an option, but just don't do it like this, because there are questions. And especially with patients in the UK, we've seen a pronounced nocebo effect. So patients who have the cheaper one, who have to change and who have an unclear test result, I mean, how are they really supposed to react? BTK inhibitors, I don't know. This is the German saying, this die Kuh vom Eishorn. Die Kuh ist noch nicht vom Eis. However, I'm grateful that we also do have some approval changes that really help us to treat our patients, also the vulnerable patients, in an easier fashion. So ocrelizumab has a different window now with pregnancy and breastfeeding, and also for cladribine tablets, there's an increased and changed labeling for active instead of highly active patients. And with this, I end 5 minutes before my official time.